My promise is I'm going to have three topics. And the first is a single slide. And it's about what I think are sort of the key three skills that innovators and inventors really need to develop. And in fact, I make the same claim of just about all young people need to develop those skills. And then I'm going to do three slides on first-in-class products and that I've worked on, products and services. And those three slides are each on sort of the underlying mechanism by which they came about. And you'll see what I mean by the mechanism. There are three that I think are important. And then I'm going to do something a little bit different. And it's really the topic of the title of this slide is that, first-in-class products and services are all that we talk about at MIT. But what many of my colleagues and students don't realize is that a lot of those new classes of products need just every bit as much innovation on the product as they do on the development. I mean, the development itself needs a new look. And I'll use some examples. Pretty much all the examples I'll use are in medical devices or pharmaceuticals, because that's sort of the nature of my research right now. And then I'll wrap up. And so that's sort of the promise for my talk. So the three key skills. As I said, I've had a chance to work on innovation for quite a while and work with a lot of great inventors. And I've had a chance to meet many great inventors over the course of my career, 35 years at MIT. And there are three things that they have, the three skills that they have developed, and that are, I'm sure every inventor that have met has these innate skills, not innate skills, there are things that they've learned. And the first is curiosity. And everybody says that, but what do we really mean by curiosity? And my claim is that curious people are collecting solutions to problems that they haven't encountered yet. So I think students and employees, it's really important to encourage them to develop curiosity, because you never know where you're going to draw from to provide a solution. The other one is empathy. And this is something, it is a skill. I mean, we all know people that seem to be more empathetic than others, but practice getting into the shoes of the user. As engineers, we talk about this all the time, but it really does take practice to see the world from somebody else's perspective. And if you don't do that, you make a lot of mistakes. And I've personally made those mistakes. I haven't realized the importance of my invention until it gets into the hands of the user, and then I find out really what it's important for. That's not the way to develop a product, right? You should know what it is. And then finally, leadership. And this is another skill that I think all students and employees need to be able to practice. And that is no one of us has all the skills necessary to develop a product. Most companies understand that and build teams this way. They generally, when they take on new employees, they hire young people from MIT or other schools. They haven't learned that. And so being able to practice leadership, convincing others to help them with the problems that they face. And I like this last one. The important aspect of good leadership is this infectious enthusiasm that leaders sort of have. And that's what I mean. It's not being overconfident. It is confidence with objectivity. All right, so now I'm going to go into the three slides about the mechanisms by which new ideas come from. And as Sherry mentioned, over 30 years ago, I was part of a small team here at MIT that worked on 3D printing. It was one of the four, depending on how you count them, there's basically four types of 3D printing technologies, and we were right there at the beginning. It was a very small world of people that were working on this. It was a fantastic experience for a young scientist and engineer, and over the course of my career, I've got to see not just the technology being developed, which is still used all over the world, but a whole industry being created. And it took 30 years. It really did. So this was in the mid-'80s, and by the time the mid-'90s rolled around, both Ellie Sachs and myself were realizing we had 12 licensees. They all had professional engineers. And they were, and we had just graduate students. Not just graduate students, but our objective in graduate students is taking them, them not really knowing anything to knowing something, being professional. And the companies that we had licensed all had professional engineers, and so how are we going to compete with that, our own licensees? And so Ellie, his real home was in photovoltaics, and so he got more interested in that, and I didn't know what I was going to do. I mean, of course, we had students, we finished up the students, but about 15 years into it, we realized that, hey, we had to move on. And for me, it turned out that here we were rapid prototyping components, and through a consulting gig, I realized that, hey, we should be rapid prototyping drug formulations. Long story how that happened. But at that time, there was an emergency in the industry, an antiviral compound that was key for the HIV cocktails that became so significant, ended up having a manufacturing problem, ended up having a formulation problem, had to be withdrawn from the market. And all of a sudden, it became, the whole industry realized that they had to have a new way to find optimized formulations that were stable. And it was a pivot for me. I mean, I went from prototyping objects to prototyping formulations, and it turned into this company, Transform Pharmaceuticals, and which was basically high throughput formulation development for the drug industry. And it was not only, well, it was ultimately acquired by Johnson & Johnson, but it was part of the change in the industry. Now every pharmaceutical company does some form of what we were doing in that company. And they hired lots of material scientists across the globe to help support that formulation development. So pivot, it's a bit opportunistic. So having an opportunistic slant on taking what you know and applying it in a new area. Adjacency, somewhat related. So I was a founder of a company, T2 Biosystems. I am a founder of a company, T2 Biosystems, that does molecular diagnostics for infectious disease. The technology involved NMR. So it was basically, I was building really small NMR machines, and that would go onto a deck. And it was combined with PCR, it made for an extremely unprecedented sensitivity for whole blood PCR. And, but I got good at NMR. And I realized that, in fact, one day I stuck my finger, I had a graduate student in the lab, and I said, could I stick my finger in one of the NMR machines and we'll take a measurement? And Chris Vasiliou, who's the graduate student, and I sort of said, what does it mean? I mean, what did I learn from doing that? And it started me, and Chris actually, on a path of trying to apply this to measuring hydration status, which today is we've done now clinical studies, and actually we believe we have the first quantitative assessment, non-invasive quantitative assessment of just how much water, excess water or dehydration is in a patient. And we're doing this now in the clinic, and it's turned into a small company called Metrites. So that's one adjacency. Another adjacency is to take, we started studying the materials behavior of how materials would change their NMR signature in the presence of other conditions. And we discovered that medical grade silicone absorbs a significant amount of oxygen, molecular oxygen. And molecular oxygen is paramagnetic, you remember from your freshman chemistry days. And that ended up being a vehicle for us to use this, a small amount of medical grade silicone to tell us what is the oxygen tension, the oxygen content of tissue during MRI-guided procedures, which is a growing procedure. So you can see it turned into Strategen Bio, which is just off the ground. And you can see there on the right is the first subject done in an MRI-guided procedure with this material on board to measure the oxygen tension in a tumor, as it turns out. And that's useful for radiation oncologists. And then finally, there's theory of change. In this case, my theory of change has been single compartment delivery of drugs and diagnostics. And that's turned into many different projects over the years. I just list two of them here. One is drug delivery to the bladder. This turned into a company called Taurus Biomedical, which was acquired 2 years ago, almost 2 years ago by Johnson & Johnson. That's doing oncology, specifically bladder cancer. And it's astounding results. I mean, it's already changed the lives of people that have been in these trials. And then the other end is still in the lab type research of delivering drugs to small regions of the brain. So we've discovered over the past 10 years that really small catheters, like as you can see here, this is something like 80 microns in diameter, 60 microns in diameter. Those types of catheters are of the scale of the cells in the brain. Actually, they don't produce any inflammatory response that it's very difficult to measure. And so you can put these things into the brain and actually use it to deliver drugs into a cubic millimeter of the brain. Instead of everywhere in the brain, small regions of the brain. And why this is important is that targeting the brain, people have thought, well, the entire brain compartment, but a lot of the receptors that you're trying to target with a drug are prolific. They're everywhere in the brain. And of course, the brain its function is very heterogeneous. So really what you want to do is not deliver it everywhere, but to put it at a small region. So in this study, we put it in just the substantia nigra on one side of the animal. And just treat that small region, it's like a cubic millimeter, and the animal's behavior completely changes. We're starting an experiment at the University of Utah this weekend on treating focal lesions for people with epilepsy. So rather than trying to do resection, which can only be done on a very small fraction of patients, we can actually deliver a drug just to the lesion for the epileptic patient. Gives you an idea of what we're doing here. So theory of change, all based on the idea that, hey, we could deliver drugs physically to just the compartment of the disease. Now, the last part of my promise, which I'll spend the bulk of the rest of the time on, and that is innovation and process. And over the course of my career, I became so interested in this, primarily because I realized that these early stage ideas at MIT could fall flat on their face if I do the wrong thing in development. And a lot of what I learned was that I should have thought about these things when I had the invention. And I became convinced that we should be teaching that at MIT. So HST 962 came out of that. And I wanted to reproduce my own education for these students in my class. And how my education happened was I didn't take a biology class. I worked with experts. Boston is such a great place to do that with all these people in these companies and the startups that I work with. And so what I wanted to do, and of course most people would say, well, we'll invite them into class. And that's what I started thinking about. All the people that I had touched, I thought about, well, I'll bring them into class. Well, it became a logistic nightmare, and the people I wanted were too busy. So I decided I would go to them. And so I did 60 interviews with, and I would video them and then edit all that. Typical interview is 90 minutes. And I would edit it down into single ideas, a couple minutes long. I'm going to play some of these for you. And then I could curate them. Hadn't anticipated that, but now I could put together a learning sequence on reimbursement or quality systems or strategy. And I'd have 5 or 6 or 7 different people expressing what they thought was important on that topic. And it turned out to be fantastic. And now, not all of those 60 interviews ended up in the course. It's a 7-week course. But about 40 did. And then 2 years ago, it turned into a MITx Pro class that we've had over 500 people, average time in the industry, which, by the way, this class is focused on both medical devices and pharmaceuticals, because it's my belief, and software and all that. It's my belief those, while they're different in detail, they actually share a lot of similarities, and that's what we focus on in the course. And it turned out to be something's good. So the average time in the industry for these folks is about 18 years. So these are not, they're different, and the discussions that we get into are just fantastic. So if you're interested, you should take a look at it. Okay, so the brief topics for today are these: strategy, POC, outcome measures. I just took a very small sampling to go through. So strategy. And now I mentioned, so Rene Russo is going to talk about an element of exactly what I said. It's like what you should think about at the beginning, even in the discovery phase. Even when it's research, what about the end needs to go into the research? We do a lot of examples in the class about that. Whoops. No. Sorry, that was my fault. Oh, that's the wrong. Nope, sorry. God, I did it. I'm so used to doing this with a normal mouse. Here we go.
Yeah, when you're thinking about early discovery, it's really important to be connecting even that early discovery effort to that clinical need, and understanding that the clinical need is going to be a decade or more away, right? And so it can be quite challenging. I think oftentimes, our scientists would discover something really interesting that potentially had a niche, but in reality wasn't clinically feasible for a lot of reasons. For example, in anti-infectives, we in the past maybe would find a small molecule that could target a very difficult pathogen in a very targeted way. But historically, the way we've treated infectious diseases, we often don't know what we're treating, and so you need broader empiric coverage of an antibiotic early on, and we couldn't get the right spectrum, the right appropriate spectrum that would be relevant for patients early in therapy. So that took a lot of things off the table that if you hadn't really thought about, gosh, what will happen clinically and how do we treat infectious diseases, you would think it's a great molecule, we should bring it forward. So it's always important, I think, to understand the clinical setting, how a product's going to be used, what other things have to support it, like laboratory diagnostics, et cetera. Good news is the field of infectious diseases is actually changing, and we're getting a lot better at diagnostics and being able to target specific pathogens and understand what patients are suffering from early on.
Yeah, so So that's just one simple example, but I can point to many others that at least I've encountered where you really need to understand what are the critical risk points in development right up front. You need to design it into the solution. And it's so true for any regulated product, like a drug or a medical device or even an airplane, is to really understand where those weak points are, because the solution has to take those into account. Okay, so then there's POC. Now, if any of you are in the medical products area or pharmaceuticals, you'll hear the language proof of concept and you think of a phase 2 trial, right? It's your first controlled trial where you're randomizing and things like that. We spend a lot of time in this course making sure that people understand that is what you'll find if you look it up in Wikipedia, POC. But what really you need to think about is there's something earlier. In fact, your first experience in man or person needs to have some element of a POC involved. And I'll speak more about this, but let's listen to Mark Levin and how he talks about those.
So thinking about clinical proof of concept in all the companies we start, and we typically work on them inside for 2 or 3 years before we start them up, that's one of the very first things we start thinking about. And that's actually 2 or 3 years even before we found the company. So we have a lot of different criteria for what we think will be an important company that'll make a big difference for patients, create tremendous value for all the employees and the founders from a career standpoint, and then certainly shareholder value. But as we start thinking about these concepts, the first thing, or one of the first things we think about is, what does POC look like in the clinic? What will it take to design those trials? How long will it take us to get to those trials? And can we find POC in the clinic with the appropriate biomarker, with the appropriate next-gen sequencing today, or is it going to be a 3 or 4 or 5 year POC? We will typically not build companies around areas where we think the POC will take 3 or 4 years. We're looking for much more clear proof of concept in the clinic within 12 to 24 months.
Yeah, so what's different about that compared with the classic POC is that the classic POC is using outcome measures, which we're going to talk about next, but outcome measures that are approvable, that the FDA would accept, because part of what you're doing in a phase 2 trial is trying to help you design the pivotal trial, the phase 3 trial, right? And but here it's different. What they're doing here is they're using outcome measures like biomarkers and things like that. I mean, the first time I got into this, it was after we'd spent $40 million before I realized, oh, we're going to ask patients how they feel? I mean, what about all these biomarkers that we're measuring? No, that's not approvable. But what's important about the biomarkers is that they tell you whether your hypothesis is correct, whether you're having the biologic, you go into this development with an idea of how the biology is going to work, how you're going to intervene, how your therapy is going to work. And so the outcome that you use at that point, it's appropriate to do biomarkers, but it's not for the FDA. It's for you and your investors and so forth. So now let's talk about, whoops, I missed one. Yeah. So these outcome measures, where do they come from? Why are they so subjective?
So patient-reported outcome measures, which are inherently subjective because they're reported by the patient, are of critical importance in the development of therapeutics. Why are they of critical importance? First of all, at the end of the day, all we really care about is whether the treatment makes the patient feel better or function better. And of course, live longer is a third one that's not subjective, but is also important. And in fact, when you go to the FDA these days and you say, I'm trying to develop a new treatment for some condition, you cannot get the FDA to approve your treatment unless the patients say they're feeling better, functioning better or they're living longer. So that's of critical importance. Now, people often get hung up on the dimensionality between subjective measures and objective measures, and one reason is that there's an assumption that objective measures are necessarily more precise as measures than subjective measures, and that's not actually necessarily true. Sometimes the subjective measures actually can give you a more precise measure of the patient's status than an objective measure. So in choosing between a subjective measure and an objective measure, although often using both is the best approach, bear in mind that subjective states actually can be measured very precisely, and there's a whole field of measurement development that is just focused on the development of precise measures of the patient's subjective state.
Yeah, so why this is so important is because it has to do with the potential for noise. So if you know you have to use outcome measures that are extremely subjective, or that there's precedent for large placebo rates and things like that we'll talk about in the next clip, you should know that. You need to know that up front, because your therapy can't just be, in those cases where you have to use those outcome measures, your therapy can't just be a little bit better, right? Because you never be able to tell, right? So it has to be a lot better. And so that impacts the kind of research I do, the kind of discovery I do. And so understanding, the indication area, it may not be the indication, but it may be the area that you're trying to treat. We'll talk about Crohn's disease here in a moment. But understanding the area that that you're proposing to address, and really understanding what limitations the outcome measures are going to place on whether or not proving whether or not your product actually works. There are a lot of, think about the difficulties in doing this. For example, Alzheimer's disease. Why has it taken so long? There's been these models around. Well, there were two things. Nobody knew who really, when I came to MIT, the only way to get a diagnosis, the only diagnosis of Alzheimer's was at autopsy, right? So how are you going to do a clinical trial? You're going to guess whether a person has Alzheimer's or not? There's all kinds of ways to get dementia, right? And if you've got a targeted biology, you'll miss it, right? It'll just be all noise. And of course, that was happening. And so you had to wait for a diagnostic, an approvable non-invasive diagnostic that would definitively tell you who of these dementia patients actually had Alzheimer's. That took decades to build that technology before you could even begin to apply the hypotheses that people had for how to treat Alzheimer's. Now, this is a great comparison because it really says, this example really points to how you have to be every bit as innovative in development as in the upfront research. And so it has to do with Crohn's disease and ulcerative colitis. And just to set the stage here, Crohn's disease, both are inflammatory bowel-like conditions. Crohn's disease has lesions along the surface of the colon, distinct lesions. Ulcerative colitis is the whole thing is inflamed. And that difference in phenotype will tell you a lot about this discussion that J.C. Gutierrez has.
There's a black and white difference between ulcerative colitis and Crohn's, not on the disease itself, but on the measure, or how do we measure it. And it's so obvious. So in Crohn's disease, the main index that we use is something called CDAI, Crohn's Disease Activity Index. And it's a formula, it's a questionnaire based on number of times, pain, 1 to 10, times they go to the bathroom, X amount of time, blood in the stools or not. It's all about self-reporting. And then CDAI has is a good tool, but as a consequence, because it's all subjective, the average placebo rate in clinical trials is between 40 and 50%. There are trials that have 70%, 60%, 30%, brutal. So as a consequence, your phase 2 trial has to have a minimum of 300 patients. Anything that is less than 300 patients, you don't even have to look at, because with these placebo rates, and drugs are being lost there, because you might have a efficacy in response or remission that is 65%, which is a lot, but with a placebo rate of 60, you don't know where you are. Years ago, in ulcerative colitis, this is for Crohn's disease, ulcerative colitis had equivalent subjective index that was called the ulcerative colitis activity score, UCAS or something like this, until a number of investigators at the Mayo Clinic decided to introduce imaging endpoint as part of the score. And they did the Mayo index. Now, this was easier in ulcerative colitis because remember, it's homogeneous. So you go with the scope, and almost no matter where you are, you can see a difference. Whereas in Crohn's, you have to make sure that you go to the same lesion, and it's not easy when you are there. That's why it hasn't been done, although it's about to be done. The FDA has seen so many disasters in Crohn's disease that are about to introduce one. So remember that I told you the average placebo rate in Crohn's was between 40 and 50%. The average placebo rate in UC was the same years ago. Now is 10 to 15%.
So it gives you an idea of the innovation here was, well, of course, they had to have the new technology, right? But the innovation here in development was coming up with a new measurement, a provable measurement. Because hey, if you're going to do a trial with 300 people in it, you better be using an approvable measurement. So there was a lot of work with the FDA to make that happen. And then the last thing, and this has become really important, you read about it every day, is reimbursement, or market access and reimbursement. For medical products, this has become a major risk point, given that you're going to take 10 years to develop the product. At the end, you better have access to the market, which isn't guaranteed, and you better have reimbursement sufficient to pay for all the development. So listen to Sharma.
So let's talk about value-based pricing and how it is affecting drug development. And I think first thing, actually, it is making us think about drug development from very early stages from an market access and reimbursement standpoint. Just to give you an idea, 3 years ago, if you asked me, the biggest risk for pharmaceutical R&D was a lack of clinical efficacy and safety. Right? Today, there are numbers out. The biggest risk for pharmaceutical R&D is market access and reimbursement. So only 25% of the drugs, or 30% of the drugs in the last year were killed because of clinical safety and efficacy. Roughly 60% were killed because of market access and reimbursement. There are examples of drugs that got recent FDA approvals but haven't sold a penny. Okay? So value-based pricing is now becoming into a part of the product selection process, which was never the case. You again went back after, I want to get into this therapeutic area, you take 3 or 4 products and you move into development. So the way it is affecting us right now in twofold. One is very early on in the drug product selection process itself, we're able to say what kind of a differentiation would allow us to price, okay, based on where we think we'll be number 1 or number 2 or number 3, and is it worthwhile from a net present value standpoint to make a decision on even developing this product at all. So it was there before, but not as a number 1 criteria, okay? It was number 3 or number 4 or number 5. The first one is, can we be a big differentiator, and then we'll figure out how we will price it. So that was the attitude 3-4 years ago. Now it is not the case anymore. Okay, so that's number one. Number two is in the development process itself. Previously, the pharmacoeconomic type of studies and studies that actually quantify the clinical benefit in a measurable way in terms of quality of life metrics, mortality and was studied more in the oncology space, but never really, and maybe cardiovascular, but not in the other spaces. But anything other than mortality in terms of quality of life and morbidity and the ability of the patient. In other words, I'll give you an example. If the difference between an active and a placebo of one pain unit can get you pain approval from the FDA. Now the question we're asking is, how long does it take for this patient to go back to work?
Yeah. So you just can read the newspaper, the risks involved. I mean, we mentioned Alzheimer's. The first major approval for a therapy there is really faced with reimbursement issues because what the trial showed it demonstrated improvement, but the market is saying, is that enough for me to prescribe to my patients? And so there's both a market access and a reimbursement problem that Biogen will have to address. I'm very hopeful that they will, because I do think it's a major advancement, but it's going to be a tough road. And this is after, I don't know what they spent, but it's a better part of $1 billion, I'm sure, to get to this point or more. And so this is what we're faced with in the industry, I think. So I'm going to give a couple minutes for questions, but here's sort of the mini summary. The first is that there are skills that we all need to practice. I continue to practice these myself. Be curious. Investigate things that you have no observable reason or no known reason that you should be looking at. That's part of being curious. Practicing getting into the shoes of other people and it's one of the reasons why diverse teams are so important. It helps you do that. It helps to have other people that have different perspectives there to help you think through what it's like to be a user, a customer. And then finally, this leadership thing is something we all need to practice on. I think this idea of pivot, adjacency, and theory of change is that everyone should have elements of all of those when they look for new products and be innovative. You have to think, one day to the next is, how can I pivot from what I'm doing? What are things that I'm doing now that could help in other areas? And of course, have a theory of change. Have a hypothesis of how you can make a difference and see if you can plant that flag in many different areas. And finally, like I said, I've learned a lot about myself, how important it is in development to be every bit as innovative as you are in the new product discovery, because a lot of new products that work are lost in development. And so you have to be just as innovative there as you are in the pie-in-the-sky product. Okay, so I'm happy to take questions. I think we got 5 minutes exactly.
I'll see if this is working. Please raise your hand if you have a question. I've got one, Michael. And we were talking about this before the session. It's really easy here at MIT to keep your mind expanding and to see all these things that might inspire curiosity. Do you have any advice for our corporate audience on how you do that when you're kind of with your head down working in industry?
Well, I'd say get out of your office or cubicle or whatever your environment is. And, for me, it's always been important to get out of MIT. Over my career, I spent a lot of time outside of MIT. I got into urology because I met a urologist and I started to study how a urology office works, and who's hired there, what are they doing, and how does the urologist spend their day. I can tell you the two most common urological procedures are catheterization and cystoscopy. That's their bread and butter. That's how they get, that's how they get paid. And that's, for me, that was, you know, it was an example of empathy, because when we went into Taurus, you know, my objective was, you know, there was going to be a urologist, the customer was going to basically be a urologist. I wanted to design a product, design a device that I can put it in front of a urologist and they don't need to read the instructions. They knew exactly what to do. And that shaped, that concept, you know, the user interface shaped it from the beginning, from the very beginning. We were still in animals. I mean, it was preclinical research here at MIT, but that's the basic concept started. It was deploy the device with a catheter and retrieve it with a cystoscope. And why did we choose that? Well, because those are the two most common procedures that physician did. And it was built in right up front in the discovery process. And so I was trying to, at that time, I remember doing it, practicing what I was preaching. And so anyway, that was because I was not just sitting in my office at MIT, I was going out.
Any other questions?
There's one, I can barely see you though. Yeah.
The question is about the segment on pricing and pharmaceutical products. Do you see a different strategy or ethical considerations when you price a medical product versus non-medical product?
Well, it's becoming, obviously, it's almost everyday an item with regard to products for COVID. But, you know, it's funny, when you look at how particularly drugs are priced, having seen and been part of those discussions, it's always built in as to how we can serve everybody independent of their insurance status. And so a lot of the, particularly drugs for very small patient populations, they build into it a mechanism by which everybody will get drug. And they just have to be able to understand, part of the innovation process is to make sure that everybody could be treated and you can still get reimbursed. And it's possible to do. And now, are there examples of where this has been abused? Absolutely. But if you look at like the Johnson & Johnson's, Pfizer's, and all the big companies, they do this. I mean, there's always the kind of, you know, the label price, but what people actually pay is usually very different than that. And they're sort of required to put in the real price. It's sort of like tuition. We're required to put in, MIT's looks like horrendous tuition. Very few MIT students pay that. We build into the process a way to make it so that it's not, that it's need blind.
Just quick follow-up. I mean, the power relationship between a pharmaceutical product or a drug and the consumer, the dependence that it creates is very, very different than, let's say, a car manufacturer or a toy manufacturer. I mean, do you see a role, more important role in regulation than when you determine pricing? Because you have a very, very, you know, it's many cases your life depend on it. So prices become whatever, you know, it's irrelevant.
So there's a whole section of my course on this topic. So increasingly, so if you go to CMS or Medicare, increasingly they're trying to encourage new product developers to share the risk. So what they will do is they'll say, okay, we will allow you to launch with this price, and we will reimburse that. But we're going to evaluate based on outcomes, so outcome-based pricing. And we will revisit the pricing based on how well these patients do. And that is becoming increasingly part of the paradigm of reimbursement. So there is a process by which you prove the value of your price model in the market. And that's increasingly become very important. So the developer is taking on more risk now. It's not like, okay, we're just going to make this and get a paycheck. It's like all of a sudden, you have to be doing studies to prove the outcome measures are what the economic outcome measures that you have said would happen actually happen. And it's complicated because in the United States, we have such crazy reimbursement landscape. There's not one reimbursement system, there's a dozen or more reimbursement systems. So for instance, the same product that's administered in the hospital gets reimbursed differently than if you do it in from a doctor's office. Completely different reimbursement model. So I know because in diagnostics, this is a big problem. Most patients that are hospitalized are there under a diagnosis-related group. The hospital, DRG is what we call it. Hospital is getting a fixed fee for the care of that patient. If the hospital physician needs a diagnostic for that patient, it comes out, it's not coming from CMS, it's coming out of the hospital's cut. So there's a big different incentive for the hospital to use your diagnostic test than it would be if the diagnostic test is, the same test is done in the doctor's office. Because then they can write it directly to CMS. Completely different landscape. Same product, same indication. So if you knew about this as a developer, you would, of course, see, can I develop a product for the doctor's office versus in the hospital? Now, it may not be possible for the particular indication, but you get the idea. You need to know that years ago when you first started dreaming up the product. It's really fascinating. And I forgot to mention, it was, I mentioned these 60 people I talked to. These were like a who's who list. One was former director of CMS, headed Medicare. His budget was the largest single budget in the federal government. He participated in this class. And in fact, he shared that point about new product developers who are going to have to share the risk in the future.
Yeah. We are over time,
Yep,
but this
sorry.
fellow has a question, so I'm going to let him ask the question. So, and
Okay.
quick answer.
Okay, quick answer.
Thank you, Professor Cima.
My wife says that all the time.
So you talked about the identifying risks and trying to get ahead of these things, but sometimes in that process, in that innovation process, you don't know what you don't know. You can't anticipate some of these things. So what methods or processes do you, I know you talked about empathy and trying to get, seeing things from different perspective, but what other methods have you found that have been helpful in this process?
Yeah, well, I think it's
Can you all hear
short
him?
answer. So I think the short answer is, well, there is no short answer. But one of the things that I found very helpful is to find related products. Products that are used by the same physician, the same patient, or something like that, and look at how they were developed. So for example, a great one, I mentioned radiation oncology, one of the companies that I started recently with Greg Ekian, former postdoc of mine. We looked at this is a product for radiation oncologists, but we looked at recently developed products for radiation oncology and asked physicians how they started using it. What decisions did they make in getting that? Now, it's for a different application, but same group of people making the decisions. It was fascinating to learn and we talked to 100 different people. We learned a lot about their decision process and what is important in those settings. And I say this because it's not exactly perfectly responsive to you, but you can get an idea of what you're going to face by looking at related products. And these can be very peripherally related. So anyway, that's my short answer. Thanks.
That was great. Thank you so much.